Welcome back to the deep dive. Today we're trying to do something, well, something that sounds almost impossible. We're trying to reverse engineer the operating system of the mammalian brain.
Yeah, not just the code, but the user manual. We're looking at a huge new atlas that maps pretty much every genetic switch in the adult mouse brain.
And when you think about it, the blueprint for a single cell is already mind-bogglingly complex. But this is about how that blueprint is managed and tweaked and regulated across thousands of different cell types.
Exactly. When we talk about genetics, most people think about the genes themselves, right?
Yeah.
The parts that code for proteins.
Right.
But the real magic, the source of all the complexity in an organ like the brain, is regulation.
So it's not what you have, it's how you use it.
Precisely. Think of your genome as this enormous instruction manual. Regulation is what decides which pages are open, which parts are highlighted, and crucially, which pages any given cell is actually reading at this very moment.
Okay, so our mission for this deep dive is to unpack this brand new atlas. What technology did the researchers even use to build a map this detailed?
The key is a technique called snATAC-seq.
snATAC-seq.
Right. It stands for single-nucleus accessibility sequencing. So your DNA isn't just floating around, it's spooled up really tightly around these proteins. The whole package is called chromatin.
Like thread on a spool.
Exactly. And when a part of that DNA needs to be read, that spool has to loosen up. That region becomes open or accessible. And snATAC-seq lets us see exactly which bits are open.
But the single nucleus part, that's the real breakthrough here, isn't it? Why couldn't we just, crush up a piece of brain tissue and look at all the DNA at once?
Because the brain is like a biological patchwork quilt. If you just look at the bulk tissue, all you see is the average.
You see the most common cell types.
You see the most common cells, and you completely miss the rare ones. You miss the tiny populations of, say, specialized immune cells, or these little pockets of inhibitory neurons that are doing these incredibly specific critical jobs.
And with the single-cell resolution, they could see everything.
Everything. Which is how they were able to profile an absolutely astonishing 2.3 million individual nuclei.
2.3 million. That is just, it's hard to even picture that scale. And inside these open regions of DNA, what were they actually looking for?
They were mapping what are called cCREs.
cCREs.
Candidate cis-regulatory DNA elements. A bit of a mouthful, but you can just think of them as the actual genetic switches.
The on-off buttons.
The on-off buttons, the dimmer switches. You've got promoters that start the process, enhancers that crank it up, and silencers that shut it down. They are the keys to a cell's identity. And this study found about 1 million of them.
Okay, so this deep dive is all about how this monumental data set, pulled from, 117 different parts of the brain, how it completely rewrites our map of the brain's internal circuitry. All right, let's talk about what that scale really unlocked. They didn't just take a few samples. They covered 11 major brain regions, from the cortex all the way down to the cerebellum. They were clearly trying to capture the full picture.
Oh, absolutely. And that rigorous approach was key. They combined that accessibility data with another huge study on RNA sequencing, which let them confidently classify every cell they found.
And we're not talking about just finding broad groups like neurons and glia.
No, not at all. They got way more granular. They delineated 1,482 distinct cell populations or subtypes.
Wow.
And then they grouped those into 275 subclasses. And get this, 253 of those were different types of neurons.
Right.
It's a new level of detail.
And that detail really exposed a massive blind spot we had. You said they found 1 million of these cCREs, these switches, but they added over 446,000 new ones.
They almost doubled the existing catalog for the mouse genome.
Which basically proves that the old methods, the bulk tissue analysis, it was kind of lying to us, right? It was hiding all this complexity.
It was smoothing it all out. And look where these million switches are. About 2/3 are where you'd expect in these intergenic and intron regions.
Okay.
But what's really significant, and you should file this away for later, is that nearly 23% of these switches are sitting inside what we call transposable elements, Ts.
The jumping genes.
The very same. And that number, 23%, is going to become very, very important.
And the power of doing this cell by cell is just so clear in their data. They found that nearly 44% of all these switches didn't match up with anything found in the older bulk studies.
It means they're hyper-specialized. More than 62% of them are active in fewer than 10 of those subtypes we talked about.
Fewer than 10 out of nearly 1,500.
Yeah. And get this, almost 1/5 of them are only accessible in one single cell subtype.
So if a cell's identity is defined by which genes it has turned on, and those genes are controlled by these switches, finding these incredibly rare specific switches is basically finding the source code for what makes one very specific neuron in the hippocampus totally different from its neighbor.
You're mapping the identity of every single actor on the stage. And this atlas is the foundational script for all of them.
Okay, so we have a list of a million switches. We know where they are.
Hmm.
But a list of parts isn't enough, right? We need the wiring diagram. We need the gene regulatory networks, or GRNs.
Exactly.
How do you even begin to figure out which specific switch controls which specific gene, especially if they're far apart?
So they used some really clever computational modeling. This is a tool called cCRE they used, and the logic behind it is actually pretty elegant. The core idea is this. If two different regions of the DNA open up at the same time in the same cell over and over again, they're probably working together.
They're co-conspirators.
Exactly. They're collaborating. So they scanned for this co-accessibility between switches, even ones that were up to 500,000 base pairs apart.
500,000. That's a huge distance on the chromosome.
A massive distance. And then they correlated that joint activity with the expression level of nearby genes. If the switches open up and the gene's activity goes up, you've probably found a link.
And the result of all that is a giant map of connections.
A massive one. They found 613,485 positively correlated enhancer gene pairs.
Wow.
The average distance for these links was about 133,000 base pairs.
Oh.
So this is it. It's a quantified, comprehensive wiring diagram for the entire mammalian brain.
So what about the master keys, the transcription factors, the TFs that actually flip these switches? Did they find TFs that were, generalists, or were they all specialists?
It was a real mix. They found some generalists, things like JUN and FOS, which you see pretty much everywhere. They likely control broad, basic cellular jobs.
Housekeeping stuff.
Sort of. But the real insight came from looking at the specialists and specifically the patterns or motifs in their networks.
And this is where it got really surprising for me, because the focus shifted to the non-neuronal cells.
Yeah, you'd think the neurons would have the most complex wiring diagrams.
Right. But the study showed that non-neuronal cells, so glia and these things called border-associated macrophages, BAMs, they actually had more of these complex network patterns than the main glutamatergic and GABAergic neurons.
Wait, hold on. The immune cells and support cells have more intricate regulatory networks than the main signaling neurons. That feels totally counterintuitive. Why?
Well, look at their example with the BAMs. In their regulatory network, they found this specific regulated triple key combination. It's a motif involving 3 transcription factors: ATF3, KLF4, and TALO1.
Okay, so what do those do?
Well, that's the story. ATF3 is an inflammatory mediator. KLF4 helps manage how cells differentiate. And TAL1 is tied to immune cell proliferation.
Ah, so you have this 3-part security system. Inflammation, specialization, and proliferation, all working in concert, just in these specific immune cells at the border of the brain.
Exactly. It shows how incredibly sophisticated their job is. They have to constantly monitor the brain's environment and be ready to respond in a very precise way.
What I find fascinating here is what that complexity implies. If the support system, the glia, the immune cells, has this incredibly intricate wiring, it suggests their roles in brain health, in responding to injury, maybe even in driving neurological disease, are governed by genetic programs that are way more complex than we ever gave them credit for.
It kind of shifts your focus, doesn't it? Maybe a huge part of the brain's computational power lies in its support crew, not just the stars of the show.
Let's shift gears now to evolution. The researchers took this huge mouse atlas and compared it to an existing one from the human brain. They wanted to see, what's shared and what makes a mouse a mouse. And this is where you get this classic structure versus function question. So structurally, they found that 58% of the mouse switches, the cCREs, had a matching sequence in the human genome.
So the DNA letters themselves are pretty similar in more than half the cases.
Right. They're orthologous. But here's the twist. They then checked if those matching regions in humans were actually used as switches. Were they accessible? Was the chromatin open?
And was it?
The conservation just plummeted. It dropped to only 22%.
Whoa. So let me get this straight. We have the same physical switch in the wall for more than half of them, but the decision of whether to flip that switch on or off in a given brain cell, that has changed dramatically between us and mice.
Precisely. The 22% that are conserved tend to be the really fundamental ones, like promoters that are just always on in lots of cell types. But the real story, the interesting part, is in the 42% of mouse cCREs that have no structural match in humans at all. the truly mouse-specific parts.
And here's where it gets really wild. These mouse-specific switches are packed with transposable elements,
Junk DNA.
the so-called junk DNA, these jumping genes that we used to think were just genomic parasites.
Yep. And the highest concentration of these TE-based switches wasn't random. It was found in 22 specific subclasses of cortical excitatory neurons, mostly in the cortex and the hippocampus. They even gave them a name, the high-E-glut subclasses.
Okay, so why? Why would this supposed junk be clustered in some of the most complex, most important cells in the brain, the excitatory neurons?
The data gives us the answer. They looked at what genes these TE switches were controlling, and they found that the TE overlapping cCREs were significantly linked to genes involved in, get this, synaptic function and synapse organization.
No way. So the junk DNA is actually running the show when it comes to how neurons talk to each other.
It looks like it. Ts aren't passive junk at all. They've been repurposed, they've been co-opted as new, species-specific switches to fine-tune the machinery of neuronal communication.
That suggests TEs are this powerful engine for evolutionary novelty.
I think that's exactly right. They're actively shaping regulation and chromatin structure, and in doing so, they could be driving the evolution of new and diverse neural circuits that are totally unique to the mouse. We're talking about over 1,300 specific TE switches directly linked to synaptic genes. It's probably the most powerful argument I've seen for junk DNA actually being an engine of change.
It really makes you wonder then, if TEs are driving specialization in mouse neurons, what could similar elements be doing to create unique human brain functions? Especially given that huge regulatory gap you mentioned.
It's a huge open question.
The sheer scale of this atlas doesn't just let us look backward at evolution, it lets us predict the future. The last part of the study used a deep learning model to see if it could predict, just from the raw DNA sequence, where these switches would be.
This is the ultimate test of whether you've cracked the regulatory code. Can an algorithm, looking only at the pattern of A's, T's, C's, and G's, figure out if that piece of DNA will be an open switch in a specific kind of cell?
And they used a model called Basenji for this.
Yeah, they adapted it for this task. And when they used it to predict the mouse results, the performance was incredible.
How
It
incredible.
was extremely accurate. It successfully predicted open chromatin across 221 different subclasses with really high fidelity. We're talking it was basically right over 85% of the time.
So the AI learned the mouse's regulatory grammar perfectly.
It did. But that's not the real payoff. The mind-blowing part is the cross-species leap. Remember, this model was trained only on mouse DNA.
Right?
The big question was, could it work if you fed it human DNA sequences and asked it to predict human cCREs?
Yeah, could it?
Remarkably, yes. When they tested it on human DNA, trying to predict the switches in matched human cell types, the mouse-trained model showed really strong predictive power.
That's amazing. So even though the actual activity has diverged so much, that 22% number, the underlying language, the syntax of the genetic grammar, is conserved enough for an AI to read it across species.
It seems so. And this is a massive step forward for modeling human diseases. Well, the study says it perfectly. This opens a window to evaluate the influence of risk variants across corresponding cell types in the human brain.
So if you have a patient with a tiny genetic variant linked to something like schizophrenia,
Instead of just knowing it's a risk variant, you can use a model like this to predict, with high accuracy, which specific cell type's regulatory network that variant is most likely to disrupt. Is it an oligodendrocyte? Is it a specific inhibitory neuron in the prefrontal cortex? It moves us from guesswork to highly precise prediction.
and you can do that without needing to get a physical brain tissue sample, which is obviously incredibly difficult.
Exactly, it's a huge leap.
This deep dive has just been incredible. We have an atlas of 1 million genetic switches, and nearly half of them were invisible to us before now.
We have the wiring diagrams, the GRNs, that show how even the brain's support cells, like macrophages, have these unbelievably intricate control systems.
And we have this completely new appreciation for transposable elements. This junk DNA is actually a powerful force driving evolutionary diversity in the most important neuronal circuits.
It's a foundational resource. It's the roadmap that's going to let researchers finally link a specific genetic variation to a specific cell type function. It really lets you stop guessing and start pinpointing where the problem is.
So to wrap this up, given that this deep learning model, trained only on mouse data, can successfully predict human regulatory activity, and the paper itself suggests using this to evaluate human genetic risk, What do you see as the biggest challenges in actually using these kinds of cross-species models to understand personalized risk in real human patients?
I think the biggest challenge is, and has to be, validation. And we know there's only about 22% functional conservation. The model is making a fantastic prediction based on a conserved grammar, but that prediction still has to be rigorously tested in actual human systems like cell cultures or organoids.
So the model gives you an incredibly strong hypothesis, but it's not the final answer.
Exactly. We're moving toward a future where we can predict who is at risk and why with stunning precision. But we have to be cautious and remember that the model is a guide, not gospel. There's still a huge amount of regulatory landscape that's unique to us.
It certainly gives you a newfound respect for the hidden complexity tucked into every chromosome and every single cell in your head. Okay, let's unpack this further next time.